Welcome to the Biotech Hangout. I'm here with John, who's the founder of Cambridge Genomics. It's a startup based in Cambridge in the UK who's personalizing cancer treatments based on a mix of next-gen liquid biopsy, genomics, and then AI built on top of that. So thanks for joining, John.
You're welcome. Yeah, it's good to be here.
Great. And so, John, we met when you pitched at Refresh in Berlin last year.
Yeah,
it was roughly one year ago. So, and you did a great pitch and really interesting company, and seems to be developing well. So, how has it been doing since you last?
It's been doing great. Yeah. So we did our fundraising over the start of last summer. Went through a program called Y Combinator, which was really kind of very transformative for us. And now kind of focusing on hiring really good people and building out a great team and actually building a product, which is very nice.
Sounds great. I will get into more details on everything you just mentioned. I guess to start with, let's say Y Combinator. I think one of the very few biotech startups from Europe who join Y Combinator. Actually, I don't know any other out of my mind right now.
Yeah, there are a couple of others. There have been others.
And why did you choose to join, and how was it? Can you just give a bit more?
So I think we were really looking for. So I mean, for anybody who doesn't know, Y Combinator is a startup accelerator based in San Francisco, and it was actually
the best in the world.
The best in the world, and actually the first in the world as well. So we were kind of looking for a way to accelerate the business, really. So accelerate kind of our development and raise some funding and try to kind of understand how to phrase our company in a way that was appealing to investors. Economics was fantastic. So I would quickly say that it's not traditionally, it's been a tech focused accelerator. This year they've launched YC Bio, which is a biotech focused accelerator, which is run by a few people, but especially a guy called Diego Rey, who sold Gene Weave or something to Roche. He's a really good guy. So they're now actively searching for biotech companies.
Yeah, that's great. I mean, it's something we are very interested in, the trend from tech VCs or tech accelerators going into biotech. And I mean, in Silicon Valley, it's already pretty advanced. You have also like Anderson Horowitz was a biofund and Funds Fund, etc. And I think you're really in the perfect space of using biological data, putting on top of that digital expertise. Yeah, it's, I guess, a perfect case for them. So it makes super sense. And so I guess YC also on the pitch day or demo day, it helped you raise your round. So can you tell a bit more on who was investors, or like how big was the round? I don't know if you published the entire round actually.
Yeah, we've not published the entire round yet. We will. So I think our first investor was a private hospital called the Comprehensive Blood and Cancer Center based in Bakersfield in California. And because that was our first check in the door, that was really helpful in kind of going to tech VCs and saying, look, it's okay. The science side is fine. People who know lots of science are okay with the science. Let's just focus on the tech side. So that was really useful. And then we went to a couple of the kind of normal seed stage VCs, so Soma Capital, Refactor, people like that. AME Ventures, Jerry Yang's fund, and various other kind of small, like strategic angel checks.
So quite a lot of US investors at the end.
Mostly yes, yeah. A couple international, including one from London, and I think that's the only UK one.
All right, that's good. So now let's switch more to the science or the problem you're trying to solve. I mean, obviously, it's like about improving the response rate to cancer treatments. It's about diagnosing cancer earlier. It's obviously one of the big untapped field where you could improve a lot. I mean, cancer treatment at the end, but I mean, a lot of people are going after that as well. So it's a quite crowded field. So,
so the only thing we care about is coming from the philosophy that cancer changes, right? So cancer evolves. your tumor today is not going to be the same as your tumor in 3 months after treatment with targeted therapy or whatever. So based on that philosophy, we think that it's a good idea to take multiple samples, multiple time points to get a kind of different dimension of the in kind of understanding of what cancers are doing. And the best way to do that is to take liquid biopsies, to take a blood draw, sequence, look for tumor DNA, and that's what we do. So really, we're very much in treatment monitoring, tumor evolution.
Yeah, I mean, liquid biopsy, I guess it's also the other holy grail that I guess is not that easy to do. Thanos can confirm, I guess. But so how are you able to do it with liquid biopsy when others like Foundation are doing it with proper biopsies?
So I mean, you can't take multiple solid biopsies really. It's pretty invasive. Well, generally anyway, it's pretty invasive. You can't puncture somebody with a needle kind of every month and sequence a solid sample of tumor. So liquid biopsy is really the only way of doing it.
Yeah, but I guess my question is more like, why is your tech? Why are you able to do it on liquid biopsy when others are doing it on solid biopsy? I mean, obviously they would like to do it on liquid biopsy as well. I mean.
So what is happening with solid biopsies and liquid biopsies is what we kind of term tumor profiling. So you take a single biopsy, single time point, and you sequence, and then you use that information to guide some targeted therapy or whatever. Nobody's really doing multiple solid biopsies over time. And the reason that we can do it, I guess, is because, I mean, basically, if you take more than one sample, more than one time point, you stop treating these time points independently, you start treating them dependently, and that means you can do some really interesting machine learning stuff on top of the information that comes out of liquid biopsy.
And I guess you can cross the different liquid biopsy of different patients with the time frame, and you can start doing some good.
Yeah, you start getting a very interesting data set. Yeah, very interesting.
That sounds, yeah. So I see the point. And then, but I guess one of your main challenge will be also access to the patient data, or to patients, or to the biopsy itself. So how do you do that?
Sure. So our first investor, as I mentioned, CBCC, they were really transformative for giving us access to patients. So we do a lot of work now out of the Bakersfield clinic. We also have 2, maybe 3 very large partnerships that we're going to announce over the next kind of month or so, with access to different huge patient data sets across the world.
And so do you do you handle the liquid biopsy, or you just get the results in and then you exploit the results?
So currently we scale almost entirely like science market, which are kind of like, they connect scientists and pharma companies with materials and with CROs and that kind of thing. And that allows us to do next generation sequencing without ever actually having to touch the sample, if you like. Okay. So that's how we can.
So you have the data set coming in, and then you exploit it and give back the feedback or the recommendation.
Sure. Yeah.
That's like, because I mean, with solid biopsy, I guess like Foundation, they are handling actually the biopsies and doing the sequencing on site, and then it's way heavier. I mean, it's way heavier as a business.
I mean, it is a kind of integrated platform where we kind of outsource the actual sequencing, but everything, it is still done under the umbrella of CCG.
Okay.
Just we use a CRO partner to scale.
Oh, I see. And so when you talk about AI, obviously it's good to do fundraising right now. I mean, everyone wants to invest in AI, especially in the Valley, I guess. But I mean, it's quite a big buzzword as well, where everyone just uses it, and it's always hard to know really what's under it. So in your case, like, how much of it is really just processing a lot of data and having a large data set, and then just combining it, versus really machine learning and like more complex AI? Like, can you give an idea of the?
So I mean, we are entirely machine learning, entirely AI. So we try to kind of see, because we're dealing with this different data set, which is, as I mentioned, kind of dependent samples. We try and see where machine learning and where AI can be useful for that specific type of data set. So we are kind of very, very heavily involved in machine learning and AI, I guess.
Yeah. Okay. So most of your team are developers, actually.
All of our team are developers, apart from me and Nimesh and Hannah.
That's good.
So yeah, we're a very tech-heavy company.
I mean, it's good, and I guess it's also the best way to make it scalable as well.
Yes, exactly.
And I guess that's my other question as well on how mature is your tech and how scalable is your tech right now? I mean, you mentioned you're working a lot on the product, so how would you? Yeah, how mature is it?
So we have an MVP which is due to be released over the summer at some point.
So minimum viable product.
Sorry, yeah, minimum viable product, which
means which is what?
It's like the very first thing that actually works and doesn't break that you can offer to people. And so what we'll be doing with that is a lot of A/B testing, a lot of figuring out what kind of information clinicians actually want to hear about the tumor, what is actually useful, and using that to kind of inform the direction of kind of where we're moving. So it's a kind of milestone in the development pathway, I would say, rather than a product which is kind of over the counter available to buy. But we will hopefully be signing some deals based on that.
Right on. A/B testing, it just
A/B testing is when you
I know what it is, and we are doing it a lot, but on testing buttons and testing parts of the website, and we don't really care on like giving recommendation A or B. I mean, you are giving.
Oh no, not A/B testing on drugs. No. Running a clinical trial without telling anybody. No, no. So what I mean is, one of the
A/B testing on the interface, not on the recommendation.
Yeah, so it's the content of the interface. So for example, one of the things we do is we give a very kind of scientific annotation of the mutations that we see in a tumor. So we use kind of cosmic annotations and various other different types of annotations. And when you speak to a doctor, they say, no, we don't really want that. We don't really want to see, unless it's really actionable, we don't really care what the mutation is doing. But when you actually show them it and show them like this, this is the mutation, this is what it's doing, they actually do like it. So I think it's important to, rather than asking people what they want directly, you show them something, see if they like it, show them something else. Yeah.
I mean, typical tech way of doing things, of showing things, getting actual feedback and actual users, and that's the best.
Exactly.
Ship fast and get feedback from the users. Makes complete sense. And so ultimately, just to understand also how you place yourself versus a physician. So physicians use it to get recommendation, or can you go even one step further of replacing, let's say, a few % of the physician inside an oncology hospital? Or like, how far can you go with the AI?
Yeah, I mean, I wouldn't like to say I'm going to replace any clinicians. That's probably not a good thing to say. I think the field as a whole will probably replace physicians or augment some physicians at some point. I don't know if we're going to be the ones to do that. I think I see this. So when we think about clinical trial design and we think about adaptive Bayesian trials, for example, in precision medicine, I can see our technology being really important for a kind of new wave of how we treat cancer patients, and that is a kind of iterative process rather than a give drug 6 months later, see if it works kind of thing. So it's much more augmenting than replacing, I would say.
Yeah, that's interesting where we're on that, on the higher perspective as well. I mean, and obviously, I guess the big challenge of precision medicine is that you have to handle so much data / different. I mean, it's very complex to set up, and it's so at the end it makes it just super cost intensive. And so if you with your tech, you can just reduce that cost and keeping the cost structure the same, and by increasing a lot the precision and the personalized, I mean, at the end the outcome is huge, and you don't so much replace the humans in it. You just really augment them. What's your view on that? On like in terms of, I don't know, time frame or how fast it will happen?
I mean, there are already some pretty kind of interesting clinical trial designs ongoing, and I'm not the best person to talk about it really. But there are some interesting clinical trials, and I think.
Do you have an example of a name? Or?
I told you I'm not the best person to talk about it. So there's I can't remember. There's one that the NIH ran a couple of years ago. Oh, there was one published like 2 or 3 days ago, actually, using HER2 antibody and lots of different patients with HER2 and HER3 mutations, lots of different cancers, that kind of thing. So people are starting to think about different trials which are not just kind of placebo arm, treatment arm, one cancer type, one biomarker. And they're thinking about ways where we can either use the same biomarker across different cancer types, or we can change the arm that a patient is in, or whatever. And I think this is coming, but as you say, I think there's big challenges in terms of scalability. So you really need, it's very cost intensive. You really, in terms of drugs and kind of active monitoring and that kind of thing, it can be very, very expensive. And I think it's really going to take and it may seem kind of cliched, but I think the technologies that are going to be really helpful for this are Internet of Things, artificial intelligence, machine learning, along with liquid biopsies, and hopefully we'll play a part in that to try and kind of scale the ability of a clinician to monitor a patient during treatment and rapidly change them between one treatment arm and a different treatment arm and a different treatment arm as their tumor evolves.
Well, that makes. I have 2 questions out of that, but the first one would be on the biomarkers themselves. How dependent are you on the development of biomarkers? Or let's say HER2. It was a huge breakthrough because it was a very clear biomarker, and you can stratify your population very easily, and then your treatment is so much more effective.
Mm-hmm.
So how are you working with biomarkers, and how dependent are you on them?
So we currently use a couple of different sequencing platforms, different technologies, and the one thing that they all have in common is they're quite kind of broad. So we're either we're not kind of following 1 or 2 biomarkers. We're following 100, 200, 1,000, whatever. So we're not super dependent on. So biomarkers like HER2 were really transformative for stratified medicine, right? Because they meant that breast cancer patients were entirely different. They were either HER2 positive or HER2 negative. Different drug, different outcome, different relapse profile, everything. I think that the biomarkers we're working with now are they're much subtler, and they're much more related to very targeted agents. So you could follow an EGFR mutation, for example, give a patient erlotinib, and then find a mutation downstream in their ATP binding pocket, know that they become resistant, switch them on to a different targeted agent for EGFR. So it's much kind of more, they're subtler, lower patient concentration biomarkers, I think.
That makes total sense. And on the liquid biopsy as well, the vision of Theranos of making it so much easier to do liquid biopsy. On the other hand, even though technically, we know all the case, but just from a vision point of view, if it was possible and reliable and cost effective, was really amazing. Mm-hmm. And on the other hand, one thing I'm also always curious is on, once you have a liquid biopsy, why not running so much more tests on one single liquid biopsy versus just doing one liquid biopsy for one test? So how do you, I don't know how you handle it, or how do you see it inside the industry of doing, let's say, one liquid biopsy, running 1,000 tests and doing that every month or 3 months, and then you have really you have data not only on cancer, on you have a lot of data.
Sure, I think the people that are the field of liquid biopsies, at least for cancer, is kind of split into early detection and kind of profiling. So obviously Grail and people like that are going after early detection, which is much more, I would imagine, going to be multi-omic sequencing, like profiling with lots of different tests. And then people like Guardant Health, who do a very kind of targeted sequencing panel, which kind of tells you what drugs are useful when you have cancer. And I think there is definitely an advantage to doing multiple multiple tests on the same blood sample, but then also, and that's kind of the philosophy that's come out of solid biopsy sampling. But one thing you have to remember is that blood is not really right. So 10 mils of blood. If you have to give a blood sample every week, it's not really the end of the world. So I think it's important to find tests which are potentially personalized, potentially targeted, that you can run on multiple different time points, because blood has this amazing property of like, people don't care about an extra blood test. They already have a blood test before they're given in a chemo infusion. Let's just take another 10 mils.
Yeah.
So that's our philosophy.
That's great. And you mentioned Grail. It's the first company that came to my mind that's kind of competing in your space. So
yeah,
is it your direct, let's say, direct competitor?
No, so Grail is, what Grail do is they try and find kind of early signs of cancer in an otherwise healthy population. And the other company that does that is Freenome. And Freenome's kind of more of a startup, but they've raised quite a lot of money now. So that is a very kind of big target. And if they manage to do it with correct sensitivity and specificity, that would be amazing. But I kind of feel like if you're screening the whole population for cancer, even if you have a 0.0001 error rate, you're still going to be telling a lot of people they have cancer when they don't have cancer, and it's going to be very expensive. So they have to be like super accurate. So
that's why they raised $1 billion.
Yeah, it's not going to be enough, I tell you.
To cover the legal costs.
Yeah, they're talking about they're talking about listing on the Hong Kong Stock Exchange, I think, as well. But anyway, so we don't really touch early detection at all. We only care about, okay, so you have cancer. Let's try and work out a way where we can give you the most effective drug, and we can make the tools that are already available and kind of help the clinician use them in the best way.
And so who is your direct competition?
Direct competitors closer to Guardant, for example, and Foundation, which you've already mentioned, people like that.
I mean, which are already very big. Like, how will you?
Yeah, they're pretty big.
How will you like be better than them, or like? I mean,
sure, yeah.
Foundation has been bought. I don't know how many, over $1 billion by Roche, and that is already really big.
Sure, yeah.
Guardant, I don't know have the details.
Guardant have raised, I think it's about either 100 or 200 million or something like that. They've raised quite a lot of money from SoftBank and others. So
how will you rival with them?
Sure. So it's really, I mean, it may seem like a small point, but it's really this kind of philosophy that tumors change, and that's what we care about. So what Guardant does is they'll take a blood test, they'll sequence it, they'll show you what mutations you have, and they'll say, okay, you've got an EGFR mutation, go on erlotinib. What we do is we take a blood test every kind of month or every 2 months, and then we model how your tumor is changing. So we say erlotinib is useful for this week, and then next week you're likely to develop resistance. When you develop resistance, we'll tell you, and you can go on to the second-line therapy. So we kind of do a much more kind of dynamic, longitudinal monitoring.
Continuous recommendation, continuous testing, but no one else is doing continuous.
So there are some academic groups. So for example, there's a big TracerX study. I don't know if you heard about it, led by Charlie Swanton with Natera, I think was the commercial partner. They did personalized biopsies, and they followed a handful of mutations over time, which is great because you can relate. You can kind of understand whether the tumor is shrinking, whether it's growing, whatever, based on these mutations. But because it was a very targeted, personalized panel, you could never detect mutations that maybe weren't detectable on day 1 and then became detectable on day 30. So our philosophy is kind of to do a kind of larger thing.
That's good. I mean, what's the number of like tumor that changes, or most of them anyway?
Every tumor changes all the time. So tumors are constantly evolving, they're constantly adapting. And when you treat a tumor with a targeted therapy, they that is kind of a selective pressure which forces evolution. So we keep talking about EGFR. It's the only one I can ever remember. So 80% of patients will become resistant to EGFR after EGFR inhibitors, non-small cell lung cancer, after 8 months, right? That is a mutation which occurred in one cell, and then that cell then expanded and took over the tumor. So the tumor completely changed in 8 months. And it was responding to therapy, and then it stopped responding to therapy. And that is happening daily.
Yeah, that makes so sense. And about responding, I guess, and one of my last question on that is, obviously, response rate is a very hot, topic and debate in biotech, especially on immunotherapies. Are you any way like also helping, or can you help the response rate in immunotherapies as well, or is it mostly linked to like immune system?
So fundamentally, we can tell whether a tumor is growing or shrinking. So it doesn't really matter if you're on chemo, radiotherapy, immunotherapy, whatever. If the tumor is not shrinking, actually the cell is not shrinking. We are looking at ways that we can incorporate some kind of interesting immune markers into our liquid biopsy tests, but I think we're quite far away there.
Right, because it's like the holy grail I hear most of the time, that even the CAR T or the checkpoint inhibitors is something like 30% of response rate to the therapy.
Yeah, and if you can find the patients,
leaves a big gap for the other people. So if you can predict on which immunotherapy will be effective for which patients over time,
especially when you start talking about treatments that cost $500,000 a year and things like that, you kind of have to know that it's going to work.
You better know, yeah. No, that's great. I think it was my last questions. Really amazing startup, amazing project. Hope you will.
Thank you.
You will go forward with that, and yeah, thanks for joining. I think you're in Boston right now.
Yeah, I'm currently in Boston. I was at a conference in Chicago, and then I went to Dusseldorf for a day, and now I'm back in Boston. So the bad thing about startups is traveling too much.
But you don't seem too jet lagged, so it's good.
Yeah, I'm okay.
All right, thanks, John, and wonderful technology. People can follow us live. I'm in Berlin, you're in Boston. Can follow us live. It's
great. Yeah, great.
Thank you.
Okay.
Have a nice day. Bye bye.
Bye bye.